Welcome to this short introduction to drug target discovery and indication expansion. If you're working in the field of bioinformatics, especially with topics like omics data analysis or network biology, you can have a major impact on pharmaceutical research. Bioinformatics is widely used to help identify new drug targets for diseases of interest and to identify new indications for existing drugs. In this video, I'll go over the terminology, try to clear up some confusion between academia and industry, talk about what's in common for the tasks, what's specific to target discovery, and what is specific to indication expansion. So let's start with the terminology. Academic research and pharmaceutical research are really two different worlds, and for that reason, it's not surprising that there's a lot of confusion over terminology. I've spent more than 20 years in academia and now 1 year in consulting working with pharmaceutical clients, so I believe I'm in a good position to help clear up the confusion. In the academic literature, if you search for drug discovery, you will find an ever-increasing pile of papers that mention the term. However, the way it's being used is as broad as an ocean and as deep as a puddle. Authors usually put the word in to try to justify the impact of their work, and of course, if you're trying to somehow link diseases and genes, it's easy to see how in academia you can imagine this allowing us to discover new drugs or repurpose existing drugs. However, unless you do more, it is really just words. Drug discovery is the very specific task of finding candidate drugs. If you're working in bioinformatics and you're not doing something like specifically molecular docking, you're not doing drug discovery. What you might be doing is target discovery. That is, try to identify proteins or complexes that are involved in a disease and where targeting them would allow us to alter disease progression, provided that they are druggable. Another term I see coming up in literature a lot is drug repurposing. It is a very broad term that generally just means finding new uses for old drugs. You rarely see this used in industry, and the reason is there are poor financial incentives for digging up old drugs and trying to find new uses for them. Instead, what you hear a lot is indication expansion. That is, you have a pharmaceutical company, they have a drug or drug under development, and they want to find additional uses of that drug to earn more money. With terminology out of the way, let's look at what's in common for the two tasks. Target discovery is all about prioritizing targets for a disease, whereas indication expansion is about prioritizing diseases for a target. This means that it's really two special cases of the same general problem, namely filling in the target disease matrix that looks like this, having all the targets and all the diseases and the associations between them. All you have to do is then, in principle, to slice columns or rows out of this matrix. The way you fill in the matrix is to take an integrative approach, combining typically automatic literature mining to capture the known targets and omics data to help identify possible novel targets for a disease. Since diseases are involved in pathways, it makes a lot of sense to combine this with protein networks, for example from the string database, to really bring it all together and find out which parts of the protein network are supported by literature and/or omics data. And that way, fill in the target disease scores into the matrix. Doing this is unfortunately a lot of work if you want to do it for all the diseases. In target discovery, we want to prioritize targets. And for that reason, if you're looking at the target disease matrix, we are interested in a specific column. However, there are three specific issues we need to address. The first is causality. We really need to find the genes that cause a disease, since targeting those is likely to net us a disease-modifying treatment or at least a symptomatic treatment, as opposed to just finding genes that are themselves affected by the disease. The way to do this is to look at genetic associations, since genetically associated genes have a better chance of being causal, and also look at regulatory networks to identify upstream receptors and kinases. Which leads me to the second issue, druggability. Whether we're trying to make a small molecule drug, an antibody drug, or some other modality, we need to be able to modulate the target with high specificity. And the reason for that is the third issue, safety. We really need to minimize side effects and toxicity. These issues arise either because of on-target effects, where you hit the right target but in the wrong place. The way you avoid that is by selecting targets based on looking at single-cell transcriptomics and looking for targets that have a narrow expression profile. The other half of the problem is off-target effects, hitting unintended targets in addition to the intended one. This is usually addressed by looking at things like sequence and structure similarity, although exactly how you go about it depends a lot on the drug modality and therefore bridges into drug design. When doing indication expansion, we want to prioritize diseases. This means that from the standpoint of the target disease matrix, we are now interested in a row. There are two specific issues we need to worry about here. The first is we are suddenly looking at many diseases instead of one disease, which means that compiling omics data by looking at individual studies simply does not scale. Instead, we have to turn to integrative databases like OpenTarget or the diseases database from my group to obtain genetic associations and literature-based associations that others have already compiled. The second issue is commercial evaluation. We want to find financially attractive diseases. This means diseases that affect many people, ideally in rich countries, and where there's not already an alternative effective treatment. The way to go about that is typically to look at statistical resources that allow us to see the disease prevalence in different countries, as well as treatment outcomes of the drugs that already exist. In addition to that, you will typically want to look at ongoing clinical trials to get an overview of the pipeline from the competitors, in other words, the competitive landscape. In conclusion, the work done in academia is highly relevant to industry, even if it's being mislabeled or oversold. Disease gene associations give us a very important common foundation for doing target discovery and for doing indication expansion. However, both require a lot of specific work before it really gets to the level where it's useful in a pharmaceutical setting. That's all I have to say about drug target discovery and indication expansion. If you want to learn more about the kinds of techniques that I'm now using in a consulting setting, take a look at this video next. Thanks for your attention.